Well, hi everybody, and a huge thank you to the organizers for the opportunity to talk with you all today. This is my first time at the MPS Society meeting, so I'm really excited to be here and have the opportunity to meet with you all. So my talk really builds on what you've heard already from Amy and Caitlin, and I'm going to be talking about how we can use genetics in the early diagnosis of MPS. So just as a quick overview, we're going to be talking more about variant classification. Caitlin already mentioned this, but particularly variant classification as it relates to MPS disorders. Why is it important? And what is ClinGen's role in this process? What types of data do we use? And how can you help with this process? So Caitlin has already outlined the process of genetic testing, but what I just wanted to mention is that variant classification is a critical part of this process. So it's one of the final stages when, after the lab has identified genetic changes, we can figure out whether or not those changes are actually causing disease or whether they're benign. We all have thousands and thousands of different genetic changes in our genomes. Some of those are benign, don't cause any issues, whereas others cause disease, and that's what we're trying to figure out here. So figuring out whether that variant is disease-causing, pathogenic, benign, not disease-causing, or uncertain significance, so we just don't know. So thinking about MPS disorders in particular, why is variant classification important? Well, I think really the major issue is that we really just want to be able to understand these genetic changes so that we can give this information to families as quickly as possible in order to facilitate that diagnosis as quickly as possible so that you can take advantage of any treatments and medical management. So whether that's diagnostic testing or from newborn screening or carrier testing, like Dr. Burton had mentioned earlier. So variant classification is one of ClinGen's interests. It's one of the major curation goals that ClinGen has. Caitlin already introduced the Clinical Genome Resource, or ClinGen, and it is an NIH-funded resource that's dedicated to providing a public resource on the clinical relevance of genes and variants. So how does ClinGen help with this variant classification process? Well, as you can imagine, there are thousands and thousands of different genes involved in thousands of different genetic disorders. And so when we're trying to classify a genetic variant to figure out whether it's causing a condition or not, what we really need to have is some expertise and knowledge and understanding of those different disorders and genes. So what ClinGen does is to bring together groups of individuals, they may be researchers, clinicians, diagnostic labs, people who really have expertise and knowledge in different disorders to come together and to create gene-specific guidance for use by that group and by the medical and scientific communities in order to help with this variant classification process. What the group then does is to classify any variants that have been reported in publications or databases. And the whole goal is really to create this ready-made, expert-reviewed public knowledge source so that the next time that same variant is identified in a patient, we already have all of that information collected together and we know, yes, this variant is causing disease, or no, it's not, we don't need to worry about that one. And also just understanding these variants better. If we know they cause disease, we can then go on and look at things like genotype-phenotype correlation. If a patient has this particular variant, what can we expect for them in the future? So on the bottom of this slide, I'm showing the process that our expert panels go through. So as I mentioned, they do create these gene-specific guidelines for classifying variants in different genes. But on the right there, you'll see that once we have gone through this process, gone through a pilot phase where we can make sure that our guidance is good and giving us the right answers, all of that data goes into public databases, including ClinVar, which is the database that Caitlin mentioned, also ClinGen's public database as well. And this process of ClinGen's variant curation expert panels, the process that we go through is approved by the FDA. So any data that goes into ClinVar from our expert panels does receive this FDA recognized database tag. So one of the ClinGen expert panels that I've been very lucky to work with, I think there are over 60 now expert panels in ClinGen, but one of the panels that I work with is the lysosomal diseases variant curation expert panel. And we've been focusing so far on genes that cause lysosomal diseases on the recommended uniform screening panel for newborns in the US. And this group is chaired by Dr. Lauren Clark, who is Professor Emeritus at the University of British Columbia, and also Dr. Catherine Rader from Duke. And we have this fabulous group of experts and also curators who collect this information, and it gets reviewed by our experts, and then we go ahead and make these different variant classifications. So we were very recently approved by ClinGen to start with our ClinVar submissions, so we can actually start sharing our variant classifications and supporting data to ClinVar. So we're very excited about that. It just happened in the last couple of weeks. We have about 140 variants classified so far, and we will continue to go ahead with that variant classification and share that data as well. So I'm going to skip over this one because Dr. Gelb already did a much more creative explanation of MPS than I have. But just to talk a little bit about the genetics of MPS, so far the one gene that we have been focusing on is IDUA, which is involved, of course, in mucopolysaccharidosis type 1. But just to note that each of the different types of MPS are caused by different genes, which have the information to make different enzymes. And so over time, we may address some of those different genes as well. But to date, all of our work has been on IDUA in terms of the MPS disorders. So Caitlin also mentioned the different types of data that we use when we are classifying a variant as either disease-causing or pathogenic or benign, not disease-causing. So I'm listing these types of data on the left. And so what our expert panel has done is to look at these different types of data and to see how we can best use that data to make a variant classification for MPS type 1. So each type of data can be used either to support that a variant is pathogenic or benign. And after we collect all of the data, we're collecting this data mainly from scientific publications and also public databases. We collect all the information together. We look at the types of evidence we have and how strong that evidence is. And then that is used to make our variant classification. So either pathogenic or likely pathogenic, meaning a disease causing, likely benign or benign, so not causing disease, or uncertain significance, which is we just don't really know. And of course, uncertain significance is really not particularly helpful when you're finding a variant in a patient and you're really trying to give them an answer for what's going on. And one of the major reasons for these variants of uncertain significance is just lack of data. We just don't know enough. So whether it's not been seen in a patient before, or we just don't have functional studies, we need more information in order to be able to move these variants of uncertain significance up to either pathogenic or benign, because that then gives us a much better answer and better information to share. So I'm going to talk just a little bit about some of these different types of data and how we use them in variant classification. I won't go through the whole list, but just to give you an idea for the types of data that we use and how that all comes together. So the first type of data that can be helpful is population data. So sometimes we look at a genetic variant and it is just too common in the general population to be causing a disease. So to think about this, you may see 1 in 10 people who are homozygous for the variant. So both copies of their gene have the same variant. And if you're thinking about this in terms of a rare disorder, let's say a disorder that has a frequency of 1 in 40,000 people, clearly this variant is just too common to be causing this disease. So immediately we would classify that variant as benign, so not causing disease. So where do we find this information about the frequency of these different variants in the population? We use a large population database called NOMAD. So the most recent version of NOMAD actually has data from over 800,000 people. So it is a huge database now. And the data in NOMAD is basically coming from sequencing of all of the genes in these individuals. So anytime a variant is identified, it would be listed within the NOMAD database, and we would know how many times that variant has been seen in different populations. So critically, NOMAD excludes first and second degree relatives because, of course, you know that you share a certain proportion of your genes with your relatives, so we don't want to overcount how often we're seeing that variant. And also in NOMAD, you would not find people with severe pediatric onset disease. So for our purposes, we use this database as basically general population adults who do not have any severe disease. Of course, they may have risk for heart disease or potentially cancer, but certainly not severe pediatric onset disease. So this is just an example showing one of the variants that we looked at from our lysosomal diseases expert panel in IDUA. You'll see here, this frequency here is based on calculations that our group did to try and figure out anything that's more common than this we would consider to be a benign variant. And as you can see in NOMAD here, the allele frequency, the frequency of that variant is much more common. And there are also over 3,000 homozygotes. So we would classify this variant as benign. So another type of data that we use is called functional data. And when we are working within the expert panel, we're not actually doing any lab experiments ourselves. We're basically collecting data from different publications and so on. But some of the publications that we would use are reporting functional data. So these publications, they can do really cool things, like they can take the actual sequence of a gene, they can introduce that gene into a cell, the gene will then be translated into a protein, and then we can measure the activity of those proteins. So IDUA, for example, we can put that sequence into a cell, it will make the IDUA enzyme, and then we can measure the activity of that enzyme. What can also be done in the lab is that they can introduce genetic variants into that gene sequence. So we can look at how those genetic variants impact the activity of the enzyme. So as you can see here, variant 1 has close to 100% activity. So we would take that as evidence for benign. Whereas variant 2, no activity in this particular assay. So we would see this as evidence of pathogenic. So our expert panel has looked at all these different studies and developed thresholds for looking at the activities that we see and whether there's evidence for pathogenicity or the variant being benign. But one of the issues we have is that historically, many of these assays that have been done don't include so many controls of known pathogenic and known benign variants. And so while we can use this evidence, we would like to be able to use it at a stronger level. So we really need larger studies with a greater number of controls. And we are aware actually of some labs that are doing this and are excited to see those reports coming out. But circling back to you all is data from patients, which is really critical, as Caitlin had already mentioned, in helping us with these variant classifications. So when we're classifying a variant and it's been found in a person, we want to know what symptoms did that person have? How closely do they match the condition that we are interpreting this variant for? And how many patients has this variant been seen in? So some of the things we think about are clinical symptoms, for example. So how closely do the symptoms match MPS 1 or what we would consider for MPS 1? Also, did that patient have particular laboratory tests, such as enzyme activity at the time of diagnosis, or what were their glycosaminoglycan levels before treatment? And then lastly, what was the impact of treatment? So with enzyme replacement therapy, for example, do we see the glycosaminoglycan levels reduce? Because with that enzyme replacement therapy, this would give us just further confidence that this patient really had the disorder that we're interested in. Also, of course, we're talking about genetics here. We are really interested in knowing as much information as possible about what was seen on the patient's genetic testing report. So clearly, we're thinking about one particular genetic variant. So what we like to know is, for example, was the patient homozygous for that variant? Did they have 2 copies of that same variant? Or were they compound heterozygous? Did they have 1 copy of the variant and a different variant on the other allele? And if they did have that second variant, what do we know about that variant? Has it already been associated with the disease or not? It's also really helpful for us to know whether parents had been tested and is there evidence that one variant came from each parent. And similarly, have other family members been tested, especially siblings, for example? Are there other individuals in the family who have the same condition? Do they have the same different genetic variants? Or if a sibling doesn't have the condition, what was their genetic testing report? What did that look like? So all of this information is really helpful for us when classifying variants. So there's a lot of information on this slide, and I apologize, but it's really just to show you that ClinGen does have an online database where we put all of this information together, all these different types of evidence, and our database will then auto-classify a variant to tell us whether it is pathogenic or benign or uncertain significance. So all of this information goes in here, our curators put the information in the database, it gets reviewed by experts in our group, and from then we can publish that information to ClinVar. So this particular variant, as you'll see, is pathogenic, and this one is benign. So this is the variant that I showed you earlier. As I mentioned, we can then send this information on its way to ClinVar. So Caitlin had previously shown an example of what a variant classification looks like in ClinVar. I'm showing you a classification here from our lysosomal diseases expert panel. It's not for IDOA. As I mentioned, our approval to be able to submit to ClinVar came pretty recently. So this is on another gene that we've worked on, GAA for Pompe disease. But as you'll see, we have this FDA recognized database tag here. As I've mentioned, the FDA does approve our variant classification process. We have the classification, which for this particular variant was pathogenic, and then all of the supporting data that we use to make that classification. So we try to make this process really as transparent as possible so that anyone can go in, they can see the evidence that we used, and even if they disagree with us, that they're welcome to, contact us. We can discuss the information, but we are really trying to make this process as transparent as possible. So speaking of ClinVar, I was interested to see what current classifications there are in there for the IDUA gene. And you'll see here that 4% of submitters, so these may be different clinical laboratories, different researchers, 4% of variants actually have conflicting classifications, which means that those submitters don't agree with each other. So one person may call it a VUS, and for another, it may be pathogenic. And Caitlin mentioned some examples of this as well. Sometimes this can just be different information from different labs. As time goes on, there could be an accumulation of further information that can help with those classifications. But more important, maybe are these variants of uncertain significance, which you've already heard about. But about a third of IDUA variants in ClinVar are classified as uncertain significance. So we just don't know whether these variants are causing disease or not. And clearly, this is not helpful for patients. So what do we do about all of these variants of uncertain significance? Well, many of these have uncertain significance because we just don't have enough data in order to be able to classify them as pathogenic or benign. And when we're looking at the scientific literature, often we just don't find the details that we need. So as I mentioned, it's really helpful for us to have as much information as possible from patients about their clinical symptoms, about their enzyme activity, their glycosaminoglycan levels, the treatments that they're on, and their genetics. Sometimes papers just don't provide that information, and that's not the author's fault. Sometimes there's just a word limit that the journal has, or maybe it's just not the focus of their particular study to be able to give all of this information, but just to say that it may not be accessible to us. And also, there are many people out there who've not been in a scientific publication, so we would have no other way to get that information. So you can help with this in that considering sharing your data. As Caitlin has already mentioned, Genome Connect is a wonderful way that would allow you to share data with ClinGen in order for us to really be able to have as accurate variant classifications as possible. And again, these are some of the types of information that we are interested in. So please feel free to come and talk with Caitlin and myself. We're happy to answer any questions. And just to give you an example of how useful this information is, I actually work with another expert panel as well for the cerebral creatine deficiency syndromes. And like you, they have a patient advocacy organization called the Association for Creatine Deficiencies. They have a registry. That registry data is shared via Genome Connect. And we have been able to shift the classification of several variants. It's just a small number so far. But as you'll see, we have 3 variants that went from the US up to likely pathogenic, and another from likely path up to pathogenic. So we're more convinced by that classification. So just really incredibly helpful to have this information for the future when these variants are seen again, when someone else undergoes testing, that information is already there and these patients can have that useful information. So just to summarize, variant classification is really important for early diagnosis, and our ClinGen lysosomal diseases variant curation expert panel has developed guidance for classification of variants in IDUA, and we may in the future move ahead to other genes involved in MPS disorders as well. Data sharing can really help to reduce the rate of variants of uncertain significance, which of course can increase the utility of genetic testing results for patients and families. And we may develop guidance for other MPS disorders in the future. And of course, again, this is absolutely, I'm just a representative of the expert panel. There are many, many individuals involved here. So just wanted to thank everybody. And again, thank you for having me here today.